All right, so I'm going to try to turn on the chat, but I will give you forewarning that I'm not very good at looking at the chat when I'm trying to go through a talk. So
I'll keep an eye on the chat for you. Don't worry.
if somebody has an important question during this talk that they can't get, they can't continue to understand the talk without asking the question, it's okay if you want to just interrupt me or put it in the chat, and hopefully Rich will interrupt me. All right, so so now I know that since I'm speaking with the neurology audience, Alzheimer's disease probably needs little introduction. We've all learned that the pathology involves plaques that consist of beta amyloid and interneuronal tau tangles. Most of us probably also understand that there's tremendous synaptic loss in Alzheimer's disease, as well as eventual neuron loss, and that the brain of patients with Alzheimer's disease has a lot of evidence for inflammation. But in spite of this incredible amount of knowledge and large numbers of people who study Alzheimer's disease, we still don't really understand what gets this whole process started. Why do people develop this pathology? And we understand that there are some individuals who have autosomal dominant forms of Alzheimer's disease, and that some of those mutations or variants that cause autosomal dominant Alzheimer's disease probably are sufficient to initiate this pathology. But the actual mechanisms of what's happening when the pathology starts really still are not well understood. So, those autosomal dominant forms of Alzheimer's disease really have led the field to what has been the dominant consensus hypothesis for really my entire career in neurology, and that we call the amyloid cascade hypothesis. And this is sort of a cartoon that illustrates the concept of the amyloid cascade hypothesis as we try to put these mutations which cause autosomal dominant Alzheimer's disease into context. And the idea is there's a protein called amyloid precursor protein, and that protein is an intermembrane protein, which is proteolyzed by several different proteolytic complexes. And one of those complexes is called the gamma secretase complex. And the gamma secretase complex is made up of a number of different proteins. The active proteolytic enzyme in that complex is either presenilin 1 or presenilin 2. And as you probably all remember, the main variants in genes that cause autosomal dominant Alzheimer's disease are in APP, the amyloid precursor protein, presenilin 1, and presenilin 2. So if presenilin cleaves APP, it can generate this fragment after also the cleavage by the base 1 enzyme that is referred to as A beta, and A beta oligomerizes to form this insoluble plaque. Now, given that this was the dominant consensus hypothesis of how Alzheimer's pathology began, one would think that we might have developed a number of different ways of trying to prevent this pathology from happening over the years. But it's not been that simple. Now, one additional piece of evidence that has developed over maybe the last 15 or so years is this accumulation of information about biomarkers of Alzheimer's disease. And this particular graph is based on data from patients who have autosomal dominant Alzheimer's disease, but you could create the same graph from looking at large populations of individuals who've been enrolled in long prospective trials of assessing them over many, many years with biomarkers, both CSF biomarkers and imaging biomarkers. And what you see is that well before any symptoms of the disease start, the first thing that happens is an increase in detectable A beta deposition within the brain, and that's done using predominantly PET imaging, as well as a decrease in the amount of A beta that you can detect in the cerebrospinal fluid. That is typically observed earliest, and then we start to see accumulation of tau within the brain and also increased tau in the CSF, and eventually the clinical features of Alzheimer's disease start to emerge. So we know that abnormalities in proteins that lead to the generation of that A beta fragment, as well as are cause for autosomal dominant Alzheimer's disease. And we know that even in patients with sporadic Alzheimer's disease, A beta accumulation probably is the first sign of detectable pathology. So why don't we have treatments that can prevent the accumulation of that A beta? And obviously, many people have heard about immunotherapy for Alzheimer's disease in the news recently, with, and this has really been in development for at least 20 years, with antibodies that try to remove A beta that has been accumulated in the brain. And we actually have had multiple trials over the years that have shown that A beta can be removed by these antibodies, yet we don't really have convincing evidence that disease, at least symptomatic disease, is influenced by the removal of that A beta. We've also had many, many trials that inhibit gamma secretase and BACE1, and none of those have had significant clinical impact. So we have to ask the question, why are these therapies not working? One potential reason is that the therapies themselves are insufficient at removing A beta. And our biomarker data suggests that they actually are efficient at removing A beta. So that's probably not the answer. It has been posited by many that one of the reasons these trials have not shown efficacy is because they're initiated too late in the disease. That by the time people are symptomatic and can be enrolled at a trial, that too much damage has already occurred. The whole disease process, the things that happen downstream of A beta accumulation have already been initiated, and blocking additional generation of plaque or insoluble A beta, or removing what exists, is not going to have an impact on disease progression. That is a very frequently posed idea about these trials, and a concept behind why we should be considering doing trials earlier. But there's another idea that is gaining some, I guess, belief behind it, which is that potentially we are not focused on the right target, that A beta itself may not be amenable to targeting by pharmaceutical or immunotherapy agents, and that we should be thinking about other targets that are more tractable as things that could be intervened on either after A beta has already accumulated within the brain and patients are symptomatic, or even things that if we could identify individuals earlier in the course of disease using these biomarkers, that could have an impact on preventing the downstream complications from developing even while A beta is present in the brain. So just to kind of illustrate this idea behind this, the changing of the timing of trials, what we currently have are many trials done at this early, at this later stage, when people are already early symptomatic with Alzheimer's disease or with minimal cognitive impairment. But it's possible that we may find that these A beta targets are effective if we can use our biomarkers and enroll pre-symptomatic individuals in these trials. There's a lot of questions about whether or not that's a tractable way of moving forward. And it would really take a big paradigm shift in the way we approach individuals as they enter midlife, and whether or not they should be screened for their individual risk for accumulating A beta and potentially developing Alzheimer's disease. I'll put in a little plug for the Duke UNC ADRC, and that concept is really one of the major themes of the ADRC, and anyone interested in ideas of what could be done in midlife for individuals and whether or not we should be thinking about screening, thinking about some of these biomarkers of Alzheimer's disease in the way we think about cholesterol, and also thinking about the things that are happening to the brain in midlife. These are all very important questions that individual investigators at both Duke and UNC are interested in pursuing. So for any trainees out there who are thinking about projects they may want to do during residency, I'd encourage you to potentially contact people in the ADRC and see if there might be ideas for your own research. Okay, so if we're going to say, okay, maybe we can look at earlier times in the life of individuals and try to do trials earlier, that's important, and I am not going to deny that it should be done. It should be done. But I also think that it's important to think about other potential therapeutic targets. And one of the ways to start thinking about other therapeutic targets is to think about genetics in AD. Now, I already talked about the autosomal dominant forms of Alzheimer's disease and the major genes that are involved in that. But what about the genetics that we have developed an understanding of from both GWAS studies and family studies that is not exactly autosomal dominant? Much of it is combinatorial sporadic genetics. And then there are also a few genes that have been identified in family studies that have very strong influence on AD risk, but are not 100% penetrant autosomal dominant genes. And so there's a lot that we can learn from all of those risk alleles that contribute to people developing either familial or sporadic AD. That's not one of the big three autosomal dominant types. And one of the things, I seem to have missed a slide here. If I can go back. There we go. So one of the things that genetics has pointed us to is that many of the genes that we have identified as associated with AD risk are involved in the process of inflammation. The risk genes are associated with proteins that are expressed in myeloid cells, some of them that are associated with familial but not dominant Alzheimer's disease like TREM2 are exclusively expressed in myeloid cells, and TREM2 is predominantly expressed in only microglia that as a cell type within the brain. Some of the other ones like PLCG2, which is really interesting, and CD33 also interesting because they have coding region variants that are associated both with increased and decreased risk of developing AD. So those are also exclusively expressed in immune cell types. And these, in addition with the main GWAS alleles being expressed in myeloid cells and increased in expression when you look at microglia from the brains of mice that have genetically engineered AD as well as patients with AD, these risk gene expression go up specifically in microglia within the brain. So all of this genetics has led to this concept that potentially the inflammatory response to A beta may be one of the major risk factors that leads to the development of AD. And one specific component of that also is the part of the microglia that's involved in the trafficking of proteins and the degradation of proteins. So you think about a monocyte, a macrophage, and a microglia. They're evolved to come in contact with pathogens, remove those pathogens from the extracellular space, and degrade these foreign pathogen proteins through their proteolytic systems. And one of the major systems in inflammatory cells that accomplishes that is the endosome lysosome system. And several of the GWAS alleles identified with AD risks are major players in that inflammatory cells' use of the endosome lysosome system to remove materials and pathogens from the extracellular space. So there's strong reason to believe that microglia and the inflammatory response may play a very important role in regulating the downstream impact of the accumulation of A beta in the brain. Okay, so what can we do to try to better understand the role of these cells, these microglia in the brain, in how they may interact with Alzheimer's disease? And one of the first things that we have to think about is what are all the different functions that microglia are doing in the brain, and could we figure out which of those functions we might want to target? So to ask that question, we have to decide, well, what are all the functions, and does every microglia in the brain have the same function? There have been a lot of different studies of microglia, and using different types of transcriptomics and even single cell transcriptomics, there are a number of different ways you can subdivide microglia, but there are at least 3 different concepts that emerge from these many studies. One is that microglia have a very important role in maintaining homeostasis in the brain. And as you can see, this particular review article, they called that the nurturer role for microglia. And when microglia are in this nurturing state, they have important roles in maintaining extracellular matrix. They have important roles in removing materials from the brain, such as myelin recycling, removing synapses during development, removing dead cells that accumulate over time in the brain, and trying to return the brain after injury to a normal state. There's also a function of microglia, very well shown in many studies, that it's that they act as sentinels. They're the most rapid sensing of brain injury. You can just do a little teeny puff of ATP in the region of a microglia and see immediate responses in their processes. And that function of microglia has been defined through, again, a lot of these transcriptional studies, because they have lots of different receptors on their cell surface, including receptors for neurotransmitters and other types of molecules that are excreted when cells are being injured or damaged. And then there's this part of the microglia that seem to take on a function of really attacking pathogens, things that we normally think about myeloid cells doing, and having this very sort of major reaction to try to wall off things like infections or areas of injury within the brain. And so you could think of that as this sort of active form of microglia. And there's always been this question, does every microglia in the brain have all of these functions, or are there different types of microglia which are primed to act in different ways? And the paradigm that I was taught when I was a medical student when I first started studying microglia was that microglia exist in this quiescent state, all of them, and then they would, in response to seeing a pathogen, go into this warrior-like state, and at some point down the road, after they'd been in that warrior state, they would transition back into this nurturer state where they would be involved in repair. The evidence for that, for this idea that every microglia is capable of doing all of these different things, is never really been very strong. It was just kind of an assumption. And more recent studies have suggested that it may not be the case that we have, one type of microglia capable of all of these different states. There may actually be multiple different types of microglia, some of which are primed to do one type of function and some of which are primed to do a different type of function. So how do we understand the role of microglia in Alzheimer's disease if we don't understand all of that diversity of microglia states within the brain? So one way that you can try to get at that question is through a technique called single cell or single nuclei RNA sequencing. Now, what you do in that technique is you would take a piece of brain, and when you use human brain, typically we have to isolate the nuclei away from the cells in order for this technique to work. And so we isolate the nuclei, we put them through a kit basically that enables us to label each active transcript within the nucleus and sequence it. And then each individual cell is barcoded so that you can put the data that you get into the barcode from cell A, the barcode from cell B, the barcode from cell C, and then you get these plots. And these t-SNE plots, as they're called, will show you all of these different clusters. And let's see if I can people see? I think I have to pick up a pointer.
We don't see the mouse pointer.
Yeah, that's
There it is. There it is. Gotcha.
Okay. You can see it now?
Yep.
Great. Now I got this captions and subtitles thing. Okay. Okay. Now it's showing everything I say. Well, that'll be in the recording. So when you get all these clusters, now this cluster might be astrocytes and this cluster might be oligodendrocytes. And if we look at the cluster of microglia, we can zoom in and see that we have a certain number of different types of microglia. When we do this on a whole brain, all of the different nuclei, there are only so many of the cells, only so many of the nuclei that are actually microglia. So we don't have a high resolution view of the heterogeneity within the individual microglia. And several studies have been published that have shown with relatively small numbers of microglia per brain, a number of different microglia subtypes. But we felt that with high resolution, we could get a better sense of the total heterogeneity within the brain and more carefully look at how AD may influence that. So we decided to take this technique in a kind of a farther step. And what we did was develop a way of enriching for microglia before doing the single nuclei RNA-seq. So in this technique, we take a cassette of medial lateral prefrontal cortex, and we do several punches through that cassette of gray matter. Then we do nuclei isolation on the tissue that we have taken from that brain. And then we label those nuclei with an antibody to a nuclear transcription factor called PU.1, which is specific to myeloid cells. Then we use a machine called a FACS sorter, which essentially is able to separate the nuclei that are fluorescently labeled from those that are not. And we isolate those fluorescently PU1 positive nuclei, and we put them through that single cell nuclei sequencing platform. And when we do that, we get predominantly microglia-like cells in our output. So it's not completely pure microglia. Some nuclei get through that are not microglia. So you can see we have some astrocytes, some neurons, endothelial, etc. But we have many, many more nuclei using this enrichment technique. And when we do that, we have, these are showing the clusters of different cell types. When we look for microglia marker genes, we get many more clusters of microglia in this technique than we do if we just look at all the nuclei. So what we did next was assemble a cohort of individuals who had a short postmortem interval, some of whom had documented no AD pathology on their brain autopsy, and some of whom really did have AD, both clinically and pathologically. And we had a mix of males and females, but as in most autopsy cohorts with Alzheimer's disease, we had predominantly females. And we had a mix of E4 allele types, with most of our controls being 3-3. And this is, again, a common distribution of ApoE genotypes in an AD and control cohort. And we had relatively close mean ages. So we did this technique on that cohort, and this was our clusters. We were able to use the clustering algorithms to get 10 different subtypes of microglia in that cohort. And when we looked at the differences between control and AD, we saw that 8 of the 10 didn't have much in the way of differences in terms of the number of cells within each cluster, but 2 clusters did show a difference. Cluster 6 was increased in the AD cohort, and cluster 10 was decreased in the AD cohort. I think I just, no, I didn't. Okay, can you still see the cursor?
Not at the moment.
No, okay. I tried to turn that closed caption off and I lost the cursor.
We can see it without the closed caption.
Yeah.
Oh, okay.
Yeah.
Okay.
Yeah, absolutely.
All right. So so here is cluster 6. When you look at cluster 6, what really came out, this is the one that's increased in AD, is that we have increased expression of all of these AD risk alleles. Not all of them. We have changed expression in many of these AD risk alleles. And interestingly enough, when you look at, for example, PLCG2, where the most common allele is one that actually decreases risk, we saw a decrease in expression of PLCG2, and others where which are associated with increased AD risk, we saw an increase in expression. And as has been shown in other studies, microglia in association with AD do appear to increase expression of ApoE. Interestingly, we also saw a big change in endolysosomal genes in this cluster 6 group. Now in cluster 10 is really interesting. This is the one where we see decreased numbers of microglia in cluster 10. And cluster 10 is really defined by expression of all these cell cycle genes. And what we saw is that there are fewer of these cluster 10 microglia in AD. So, these cell cycle genes are, they're turned on in cluster 10, but in microglia from AD brains, we don't have as many of those proliferating microglia. So what can we say about these findings about what the what's actually happening to microglia in the AD brain. So in cluster 6, where we see more cells, it appears that that particular type of microglia has a transcriptional profile that is associated with increased expression of genes associated with the AD risk alleles, but also with this endolysosomal activity. And so we could hypothesize that AD is associated with an increase in the microglia subtype that is involved in removal of foreign proteins. And that makes some sense, that microglia are either, that new microglia are either being generated to do that, or that some microglia which can transform to a different subtype may be primed to actually go in the direction of that behavior. In cluster 10, what we see is fewer microglia showing that proliferative type of behavior, and or proliferative transcriptional program. And that's probably not totally surprising also, because several studies have shown that markers of senescence increase in microglia in the setting of aging and AD. Now, we saw this loss of cluster 10 microglia even in a relative to an age-matched cohort. So we don't think it's an age effect alone. Clearly, AD is also having something to do with this decrease in the number of cluster 10 microglia. Okay, so that's the first part about what I wanted to talk about today. And so I'm going to move into a little bit of a different topic, but I wanted to just kind of summarize that what these studies showed us is that we can enrich for human microglia nuclei and use single cell RNA-seq to get a better sense of microglial heterogeneity. I didn't show you this, but ApoE status does not impact this clustering. We saw the same clustering pattern, whether we separated by E3 and E4. Also, we saw that all clusters had some detectable changes in gene expression, but the sizes of the clusters themselves were only impacted in cluster 6 and cluster 10. And these two clusters do seem to have some potential roles in AD pathology. Okay, so what we really want to know is, could we modulate microglia capacity to do the things that they need to do to potentially decrease the impact of the accumulation of A beta in the brain? And one of the things that we decided to look at, started quite a long time ago, was a microRNA. And the reason that we decided to look at microRNA is that they have a strong apical role in regulating inflammation, microRNAs. So microRNAs are these small RNAs that can, when they are generated, can influence both the stability of whole groups of other RNAs by binding to different regulatory regions within transcript and causing them to either not be translated or to be degraded. And we decided to look at a particular microRNA called microRNA 155, which had been shown by many different groups to have a dominant role in regulating inflammatory behavior in myeloid cells. And so microRNA 155 its expression is induced by common regulators of inflammation, such as TLR4 signaling. And so when inflammation happens, microRNA 155 expression goes up. After it's up, it also induces, it promotes NF-kappa B signaling, and it induces the expression of microRNA 146. A is the major one, but there's like a B and a C. And microRNA 146 actually has an anti-inflammatory effect. So microRNA 155 is the inducer, and then it induces its own negative regulator. And what we see in microglia is we can see the phases of miR-155 going up, miR-146A going up after inducing miR-155, and then the induction of eventually more anti-inflammatory behavior. And we had also shown, I don't know, 10 or so more years ago, that if you can get cells into more of an anti-inflammatory state, you enhance their ability to internalize A beta. And so we decided to explore whether or not if we blocked miR-155, we could get more microglia to be in the state of internalizing A beta and avoiding the state of being pro-inflammatory and releasing all kinds of factors that can be injurious to neurons and impair the function of the nervous system as a whole. So we decided to develop some tools to study miR-155, and this included several lines of mice. And what I'm going to talk about today, every time I get close to this is this particular tool where what we did was we put LOXP sites, and LOXP sites enable you to use this enzyme called CRE to remove a piece of the gene. And so what we did was we created a mouse where we inserted these LOXP sites around the miR-155 gene, and then we could use that mouse line to remove miR-155, and we could use cell type specific CRE mice to specifically remove miR-155 only from microglia. And what happens when we do that, and we look at A beta internalization, as we can use different markers to look at A beta internalization. So here we have green A beta that would go anywhere within the cell, and red A beta that only goes into the endosome lysosome system. And you can follow the internalization of both the green and the red A beta. And when we did that assay on cells after we either removed miR-155 or overexpressed miR-155, you can see that overexpression decreases the ability of microglia to move A beta into the endosome lysosome system, and miR-155 knockout actually increases the ability of microglia to get A beta into the endosome lysosome space. Simon has asked a really long question. Should I answer it now?
Sorry, let's wait till the end on the.
Okay, all right. Okay. So then what we did was we crossed the miR-155 floxed mice with cells that expressed CRE recombinase under a microglia specific promoter that is induced by giving the animals tamoxifen. And then we crossed that into this Alzheimer's transgenic model. And we used two different models. We used what's commonly called the APP-PS1 model or the Borscht model. And then we used another one called the 5X FAD model. And so we decided that we would see what happened when we removed miR-155 from microglia. And what we did in this experiment is shown here. These are all of our different lines in our treatment groups. And we gave the tamoxifen to remove miR-155 at 2 months of age. And then we looked at the mice at 6, 9, and 12 months. And you can see here that when we gave the tamoxifen, miR-155 expression was lost. And then this is the DNA, and here's the actual miR-155 measurements. When we FACS isolated microglia from these different groups, we saw that, first of all, that in the APP-PS1 mouse model of AD, there's a big increase of miR-155 in microglia. And when we gave the animals tamoxifen, they lost expression of that miR-155. Okay, so what happened to the mice? So the first thing we looked at is whether or not our hypothesis was true, that A beta would be removed more effectively by mice that lost miR-155 from their microglia. And we saw that that actually was the case. Looking biochemically at A beta, we saw a reduction in insoluble A beta. Looking at plaque, we also saw a reduction in plaque relative to either corn oil or treated mice or mice that didn't have the miR-155 floxed gene. And when we measured the plaque area, we saw a big reduction. So this was great. We were, our hypothesis turned out to be supported by the data, but we had a bit of a problem. And the problem was that the mice were dying, and they were dying even more than they typically die in the APPS1 mouse line. Now that mouse line does have more mortality than one would expect from an AD model. And several studies had shown that the main reason that these mice die is because of seizures. So we decided to see if the increased mortality, this is the group that had miR-155 knocked out, was due to seizures, because we wouldn't expect there to be increased mortality from A beta because the A beta had been removed. And so I apologize for the kind of low resolution of this, but I was having trouble with the file, so I had to resave it in a lower data space. But what we saw was when we gave tamoxifen and we recorded with the electrodes that were surgically implanted in the mice, we saw that there was there is some hyperexcitability in this line at baseline, but it's after we gave the tamoxifen and removed miR-155, we saw frank spontaneous seizures. And when we looked at these are the weeks after tamoxifen treatment, we saw increases in seizure duration and increases in the number of seizures. So clearly, and we even, had during these mice that were implanted with these electrodes, several animals exhibit Sudep, but basically have a seizure and die. So we were pretty confident that even though we saw removal of plaque, the mice were having worsening hyperexcitability, epilepsy, and Sudep, and that was the cause of their mortality. So the question is, why do we have worsening brain function even when we're more effectively removing A beta? And when you get back to the question about, is removing A beta going to benefit either animals or people once the process is started, you have to actually, think about the clinical trials, and maybe that's actually not going to help. So even though we used, what we're trying to do is push the cells to a more anti-inflammatory state and promote their ability to internalize A beta, what we saw was at the same time as they were internalizing more A beta, they also were changing the degree to which they were internalizing synaptic material. So what we did was we isolated the microglia from these mice, and this particular figure is from the 5X FAD mice, which we used because they had better survival, they had fewer seizures and much less mortality, but they had the same result of decreased A beta in the brain after microglia had miR-155 removed. And we saw that that there was in just the 5XCD mice, the microglia would have increased internalization of VGAT and but no change in internalization of vGlut1. And after we removed miR-155, we saw a change where the microglia were more likely to internalize these vGlut1 terminals and less likely to internalize these VGAT terminals. And it might not make complete and obvious sense why this result would be associated with increased excitability, but you have to think carefully. As studies of Dravet syndrome showed us early on, it didn't make sense why animals that had knockout of SCN1A would have seizures, but eventually, complex circuitry was determined, and it was shown that the reason is because you lose excitatory inputs to inhibitory inputs, which then actually change the overall excitatory balance and can lead to seizures. At this point, we don't know the circuitry. We've only done these studies by FACS, but our hypothesis is that clearly, even though microglia are more capable of removing A beta, they're also dysregulated in how they are removing synapses in this setting. And it's an important finding to consider when trying to target therapies at microglia and change their A beta internalization. Okay, so the conclusions from this part are that microglia do use miR-155 to influence their ability to internalize things like A beta, that you can actually in vivo make them more effective at removing A beta by removing miR-155, but unfortunately will also change their ability to target certain synapses, which can result in increased hyperexcitability and seizures. Okay, so I want to acknowledge all of the many people who have worked on these two projects. The Microglia Heterogeneity Project is really run by my longtime colleague, Suman Jayadev, and Kevin Green in her lab developed the technique for PU1 nuclei isolation. And Catherine Prater, who was a postdoc with me, did all of the analysis of the single cell RNA-seq data. We got a lot of help from Noah Snyder-Mackler's lab in developing the technique, and from Ben Logston and his group in doing the bioinformatic analysis. And Dirk Keen at the University of Washington helped us with the accumulation of that cohort of autopsy material that we used for that study. All of the microRNA miR-155 studies have been done by a former graduate student who's now a postdoc at Baylor, and her name is Macarena Aloy. She was a very, very productive and I would say determined graduate student to work with that many mice and create that survival curve. And then the rest of the members of the lab helped in the analysis of the data, and the EEG studies were done in Horacio de la Iglesias' lab. And again, Dirk's lab helped us with the A beta biochemical assays. All right, so thanks for the opportunity to talk with everyone today, and I'm happy to answer questions in the chat.
Thanks, Gwen. I'll orchestrate it.
Okay.
Pratik, do you want to go first?
Thank you so much, Dr. Gada. It was wonderful to talk, especially for people like me who are just like starting to learn about aging and Alzheimer's. So this is very helpful, and good to see the importance that well-deserved to microglia is now finally coming into picture, not only on the scavenger function, but also like startups getting funded by big pharma for transdifferentiating microglia into actual neurons, and that I thought pretty fascinating. I have a couple of questions actually for part one of your talk, more like methods question, where you talked about the single cell RNA analysis of microglia. My question is more directed towards the neuron single cell RNA analysis, and rather than getting a tissue from the brain, if there is any non-invasive method that you think is feasible, for example, olfactory epithelium scraping that will pick up some of the olfactory neurons, for example. And if so, would it be amenable to direct processing and RNA analysis, or you need to actually create a culture before and multiply those cells before you can actually do that?
Well, I'm not really familiar with how many cells that you get out of that type of a analysis. But what I would say is that you do need thousands of cells at a minimum to really get strong bioinformatic signals from this type of approach. And by thousands, typically tens of thousands are better than ones of thousands.
Right.
But yeah, so I mean, I'm not quite sure. But we not my group, but other groups have done these types of analyses on organoids. And that's another way of trying to get at some of the questions you're asking, where you can generate iPS cells and then generate neurons without having to do autopsy studies. And we like autopsy studies, and we really like autopsy studies because we actually have autopsy confirmed diagnosis on autopsy studies. And that's one of the things that's very difficult to be confident of when you're using IPS, because you often don't have that kind of clinical information. The ADRC network, there are more and more well-characterized individuals with also IPS where you have strong biomarker-based diagnoses. And so that's another alternative way of trying to approach this without having to use autopsy.
Thank you. Yeah, and a follow-up to it real quick is, there's tens of thousands of cells. Would it be all as a single population, or if it is part of the mix, for example,
Well,
other?
I mean, you get the strongest if you're going to look at one cell type within a mix, you get the strongest signal if you have tens of thousands of that cell type. And so you can get the best bioinformatic analysis if you have tens of thousands of the type of cells. That's why we turn to enrichment, right? Because normally in an autopsy study, you wouldn't get tens of thousands of microglia.
Okay. All right. Thank you.
Simon, do you want to go?
Sure. Thanks, Dr. Gunn. It was a really good talk. Really, really interesting. One of the challenges that we've faced when we're looking at single cell data is trying to identify whether cells are actually subtypes or they're different states of a particular subtype. So they're transitioning from activation, quiescent, etc. So have you looked at the sort of states that the cells are in, the microglia in within the clusters? And then the follow-up question is, have you carried out sort of cell-to-cell interaction analysis, receptor ligand interactions and things like that within your data?
Yeah, so let me answer the second question first. So this is unpublished data, and we have definitely not completed all the analysis that we want to do on those cells. So we have a couple of things that are going on right now. And since I'm no longer in Seattle, it's hard for me to be up to date on exactly where things are on the pipeline. I know that's on the list, but I don't know if it's been accomplished yet.
Right.
And I think not, but I think it's going to get done eventually. What we really are most interested in and what's going on right now that I know of is these trajectory analyses. And trying to figure out, does cluster 10 generate cells in cluster 7 and cluster 8, that kind of thing. That's what we were most interested in the as our next step.
Okay.
But you're right, the ligand receptor analyses is critically important, especially if you're thinking about eventually using this type of data for generating hypotheses about targeting. The first question, I'm going to plead a little bit, I don't know, PI ignorance, but a lot of thought. And there are actually several bioinformatics individuals, who have been asked to work on various aspects of this data. And a lot of thought with both statisticians and bioinformaticians went into the clustering. Exactly. And literally Catherine sat on this and wouldn't give me, because she knows I'm going to go talk about it, she wouldn't give me the clusters until she was confident that they were the actual clusters. So she spoke with, I think, 3 or 4 different groups that were doing single cell data analysis at the University of Washington. And so they have been thoroughly vetted as to these are the clusters. And it wasn't like when when we did the first initial studies and we saw 9 clusters, some of those clusters actually got lost in the data, but then some new clusters emerged when we had a lot more data. And then there were initially more than 10 clusters. Some of them were sort of individual specific right? So we decided not to include those 10 clusters in our further analyses because they only showed up in, one individual brain.
Yeah.
And so we did, remove some of those clusters, and other clusters were combined because of statistical issues. So I think we started out with something like 14 clusters in the whole data set, and then it got weaned down to the 10.
Okay, great. And if you ever wanted to have someone more proximal, I've got 3 bioinformaticians in my group who have been analyzing single cell data for years. So be happy to help out if you needed any.
Yeah, it would be great. We should talk sometime.
Yeah, excellent. Thanks, Dr. Garden.
Alex, did you have a question? Alex is speechless.
Maybe no longer here.
She said 2 compliments about how great the work was. Okay. Kevin.
Yeah, you used the term Sudep during your presentation for the animals with the MIR 155 knockout. That's a hot topic in epilepsy, as you know, what the EEG is. Is it brain? Is it cardiac? Is it respiratory? There are several theories that go along with that line, but was EEG available from the time of death in these animals? And
Yes.
what kind of seizures were they having?
Well, so mice, so interestingly enough, mice don't have a lot of variety of seizures, and we only have 4 leads, right? So we don't have a ton of information, but what we were able to see is that some mice, when they were hooked up, were having generalized seizures, and typically when mice have generalized seizures, they have a very characteristic physical generalized seizure that you can see. And so we saw both on video and on EEG that the mice were having the generalized seizure, and then they died. And so the generalized seizure was continuing, and then it just stopped. We don't have EEG, ECG monitoring given that setup. So we don't really know if the heart stopped first.
Right.
Because that's just the limited type of monitoring that we had. But the EEG was showing generalized seizure before the mouse died.
Yeah, and I think that's a key point. It still could be cardiac or respiratory due to active seizure activity, but that's an important feature, because the real question is this a postictal phenomena or is it an ictal phenomena? Thanks.
Yeah, as best as we can tell, the animals are seizing and then they die. Right? That's we don't have any other additional physiologic monitoring at the time.
Right. Thanks.
Kirby.
Thank you. That's a good talk. I just have a quick question. What were the effects of tamoxifen on the AD models without the knockouts?
Well, there was no effect on either the A beta measurements or on survival. We didn't look at any behavioral change.
All right.
Was that your question?
Yeah, tamoxifen is a kind of a mitochondrial toxin, so I'm just wondering.
Yeah, we use a single dose, and you gave that single dose at 2 months. We didn't look at behavior because we really couldn't. We had big plans to look at behavior, but we just had too many losses of mice to really be able to look at behavior in the whole cohort. So then we just sort of abandoned behavior. So we don't know if that single dose of tamoxifen at 2 months had an impact on progression of behavioral change. But most of these mouse models don't have huge behavioral effects from the AD model anyway. You can see them, but they're relatively
I was more
small.
interested to see if they had if the tamoxifen had a seizure eliciting effect on those.
Oh, yeah, no, we did not see that. We didn't see that the tamoxifen by itself had any impact on the seizures.
Okay, great. Thank you.
Carol, are you out there as our resident microglia macrophage expert?
Hi. Okay, good. I'm echoing. How do I fix that?
Ooh.
I can't stop the echo.
Just make your question or comment quick.
It is a comment. It's in the comments. So can you look at it and see if you can read it for me?
I don't see it.
Oh, all right. Let me just state it.
Yeah.
What outcome would you predict to surrounding neurons? Okay, can you hear me better?
I can hear you.
Okay, good.
There.
Can you hear me now?
Yeah,
I feel
perfect.
like I'm on the space station.
Yeah, I can
All
hear
right, go back again. What outcome would you predict to surrounding neurons if all the macrophages were converted to the final stage? So, you know, the three divisions here has been a traditional idea about microglia, but, you know, the ability to transition and what those transitions mean is, you know, you're exploring and we're exploring as well. And it's really very, very interesting. So if you converted all of them to the final state, what do you predict would happen to the brain?
So I mean, it really depends on what you think the final state is. So, and we don't even really, like when I say we're working on these trajectory analyses, we don't think that there is one final state because the trajectory analysis really point to a few branches. But, you know, again, that's all bioinformatics and it's hard to know what really happens. But if we'd said like, if all microglia were activated and going after a pathogen, I think that would be bad for surrounding neurons. If all microglia were in a quiescent state and synthesizing extracellular matrix, but not necessarily internalizing things, that could be bad too. I mean, we know that if you don't have functional TREM2, you know, people develop a neurodegenerative disease, right? So really, I think you have to have these balances, why there are potentially 10 different behavioral transcriptional subtypes of microglia in one little piece of medial prefrontal cortex is because you need microglia to do a whole bunch of different things. And it's probably not the case that every microglia is doing all of those things. So, you know, so I think if they were all of one type, we would be missing functions that are critical to brain homeostasis.
Oh, totally agree with that. But on the other hand, you have to understand that there's probably no terminal state, that in my belief, there's no terminal state, that it's always doing something. So unless there is a rock and the cell is dead, you know, but the anti-inflammatory state has as much power to me as the pro-inflammatory state.
Absolutely. I mean, that's what we were, and you can see that it has both positive and negative effects. And we were trying to induce the anti-inflammatory state by knocking out miR-155. And we saw that the cells were more phagocytic, but if they're not controlled in the way they are phagocytosed, then they can do, you know, both good and bad things.
Yeah, so it's really sort of interesting how those switches occur, and that's what we're fascinated with, is how those are switching. Just it's really nice to hear you working on it. It's great. Thank you.
Thanks, Carol. Gwen, thank you so much. Great talk, a lot of interest. I think this is the longest we've gone till 9:16. So everyone have a safe day. And again, Gwen, thank you so much.
All right, thanks for the invitation. It was nice to talk to you all.
Take care.
Thanks.
Bye.